Hello, my name is Erica, and today I will be telling you about Duchenne Muscular Dystrophy and the Dystrophin Isoforms. What is Duchenne Muscular Dystrophy? It is an X-linked disease that affects 1 in every 7,250 boys born in the United States. The disease is caused by a loss in the dystrophin protein in the muscle, which causes most boys to be in a wheelchair by the age of 10. The loss of dystrophin also affects the lungs and the heart, which makes the disease fatal and causes most DMD patients to pass before the age of 30. Dystrophin is a protein that helps anchor the actin cytoskeleton of the muscle cell to the external environment, which prevents tears in the muscle that are caused by contraction. This is done through an entire protein complex, and without dystrophin, the complex is lost and the muscle is prone to being torn. This causes vital muscle proteins to leak out of the cell and extracellular molecules to come in, which is devastating to the muscle cell. The complex has also been found to be important in cell signaling. The complex is not only found in the muscle cell, but also in other parts of the body in various isoforms. The gene encoding dystrophin has 79 exons. The muscular dystrophin is the full-length protein, but many other lengths exist due to differential mRNA splicing. Muscle dystrophin has a molecular weight of 427 kilodaltons and is referred to as dystrophin protein, or DP 427M, for muscle. There are other full-length dystrophin proteins, including DP 427C, or cortical dystrophin, and DP 427P for Purkinje dystrophin. These are called isoforms. There are also other smaller dystrophins like DP 260, retinal dystrophin, and DP 140, kidney and brain dystrophin. Therefore, patients with mutation in muscular dystrophin also have mutations in other full-length dystrophin proteins and may have mutations in the smaller isoforms, depending on the location of the mutation. The most commonly studied dystrophin is DP 427M, the muscle dystrophin that is found in both skeletal and cardiac muscle. As mentioned previously, patients lacking dystrophin are more prone to muscle tears. This leads to a cycle of muscle cell degeneration and regeneration, as can be seen in the muscle fibers. Healthy muscles have nuclei in the periphery of the cell and have very little fat or fibrosis accumulation. In DMD patients, the nuclei are commonly found in the center, which is a sign of muscle regeneration. There is also frequent immune cell infiltration to clean up the area and signal degradation of damaged muscle and regeneration of new muscle. Finally, fibrosis occurs when fat is accumulated and takes up the space that the muscle cells have left behind. Retinal dystrophin is an isoform that is not as well studied. It is known that at least 3 isoforms of dystrophin are found in the retina. The retina is an important part of the eye that turns light stimuli into neuronal signals that allow us to see. The retina is made of many cell layers, and the dystrophin isoforms are found in these cells are thought to help with signal transmission. DP 260 and DP 427 have been found in the outer plexiform layer, while DP 71 has been found in the inner limiting membrane and blood vessels. Some studies have found that DMD patients have poor red-green color vision and also have a loss of contrast sensitivity, which could be related to a mutation in the retinal dystrophin isoforms. Another less studied isoform is the kidney dystrophin, DP 140, which is also found in the brain. In 2013, Oliveira and colleagues did a study on mice to see if the kidney of MDX mice, the mouse model of Duchenne Muscular Dystrophy, had any differences to wild-type mice. They found that the urinary space in the glomeruli of the kidney was reduced. The glomeruli help filter blood plasma and contribute to overall renal function. Additionally, they found that the MDX mice have larger hemorrhagic areas compared to wild type, which are signs of renal dysfunction. But what does this mean for DMD patients? Most patients pass from cardiac or pulmonary failure. However, there have been documented cases of DMD patients passing away from renal failure. At the minimum, DMD patients are at risk for renal dysfunction. The state of medicine has improved and caused less patients to pass early from cardiac or pulmonary failure, and now renal failure may be another problem that is worth researching. The final dystrophin isoform we will be discussing is brain and nervous system dystrophin. These isoforms are beginning to get more research interest compared to the other isoforms discussed besides the muscle dystrophin. Many isoforms have been found in the brain and nervous system, including the Purkinje neurons of the cerebellum, which contribute to motor and spatial learning and planning, the cortex, which is involved in higher thought functions, and the peripheral nerve, although it has not been as highly researched. Research interest in these isoforms is increasing due to the prevalence of cognitive deficits of DMD patients. In particular, 30% of patients show cognitive deficits with an IQ lower than 70, and it is thought that this is an underestimate. Additionally, DMD patients have shown to have higher chances of mood disorders and autism spectrum disorders, as well as a higher chance of epilepsy. Further research has shown that DMD patients have motor and spatial learning impairments. In particular, DMD patients with mutations in DP140 and DP71 have these deficits compared to patients with mutations closer to the N-terminus. Lots of research has asked why this is the case. The answer seems to lie in the dystrophin isoforms' role in synaptic transmission. Synaptic transmission is the way signals are sent throughout the brain to other areas in the brain or the body. Synaptic transmission relies on the use of neurotransmitters, molecules from the presynaptic neuron that bind to receptors on the postsynaptic neuron that can trigger or prevent a signal to continue to the next neuron. The dystroglycan complex has been shown to contribute to the clustering and stabilization of receptors, especially the inhibitory GABA receptor. Proper neuron transmission depends on the proper arrangement of these receptors to relay the signal. Deficits in dystrophin have been shown to have deficits in long-term potentiation and long-term depression, which are important components of synaptic plasticity. And synaptic plasticity is the basis of all kinds of learning, including behavioral and motor. This is why it is thought that DMD patients experience these cognitive impairments. In conclusion, I hope this presentation has shown that the dystrophin isoforms are also very important to the Duchenne Muscular Dystrophy disease, as it has been shown that DMD patients are prone to vision problems and to renal dysfunction and possible failure. Additionally, the brain isoforms are associated with cognitive impairments, which is very prevalent in DMD patients. Furthermore, there are more isoforms in the lymphocytes and Schwann cells that we did not get a chance to talk about, but are not as well researched and studied, and may contribute more to disease progression. It is undoubtable that more research is necessary to understand exactly how the dystrophin protein isoforms and their associated protein complexes are affected in DMD in order to treat the disease and its symptoms. Thank you so much for your attention.